This is the lab lecture video for the General Chemistry 2 lab chromatography. This lab lecture video is going to be focused on the theory, like what is chromatography, how does that relate to things we've talked about in lecture, and the calculations that go with it. The lab lecture video about the procedure is a completely separate video. So this one is focused on the theory and calculations. There is a second video focused on the procedure. So chromatography. First, we need to talk about what the heck that is. Okay, well, chromatography is a super commonly used technique for chemists, and not just chemists, but scientists everywhere. Chromatography is a way to take a mixture, so a homogeneous mixture. I guess it can technically be a heterogeneous mixture, but really it's just a homogeneous mixture that we want to separate its components. And when we do chromatography, it can be done several different ways, but the two ways are qualitative and quantitative. So qualitative chromatography is our job is to take our mixture, separate it into its components, and figure out what are the different components. That's it. Just what are they? Okay. Quantitative chromatography is when we want to take our mixture and separate it into its components and also know how much of each component there is. So there's numbers that go with that. So we're going to do both types in this lab. Gas chromatography, which is the part A portion of this lab. So part A is gas chromatography, which GC is the abbreviation for that. That is going to be quantitative. So at the end of this, you'll know how much of all the components of the mixture there are. So parts, well, I guess the rest of the lab, so like B through D, are qualitative, which means we're just figuring out what is present, and we're using thin layer chromatography, so I'm combining letters, TLC, thin layer chromatography, which is qualitative. So we're just identifying what is in the mixture. Okay, so how chromatography works? Well, it's the relationship to the things that we're talking about in chapter 13. The solutions stuff are the intermolecular forces. So when we do chromatography, we have to have two different things. We need the mobile phase and the stationary phase. And with chromatography, this is going to be shocking. The stationary phase doesn't move, it's stationary. And the mobile phase does move, it's mobile, okay? And we take our solution and we introduce it to the stationary phase and the mobile phase. And the different components in that solution, although they've already mixed together, so they have somewhat similar, the like dissolves like rule, they've already dissolved with each other, but they're not all identical. And so typically, we will make the stationary phase non-polar, all right, NP for non-polar, and the mobile phase polar. Or it could go the other direction, and they don't have to be one super, super non-polar and one super, super polar, but one is more non-polar than the other. Okay, so the components of the mixture will be more strongly attracted to one of these two things, either the stationary phase or the mobile phase. Well, I'm going to use the example of thin layer chromatography to kind of demonstrate this. So in thin layer chromatography, we have a piece of glass that has been treated with a solid, which is the stationary phase. And we're going to put it in a beaker with some liquid in it. Okay, and that liquid is the mobile phase. So we'll take our sample and we're going to put it on the stationary phase and then dip it in the liquid and let the liquid rise up. Just like putting a piece of paper in water, it's going to like wick up. And as it moves up, the different components, so that blue dot would be a drop of the whole mixture. So let's say there's 3 things in there, okay? Well, those 3 things are going to be attracted to the stationary phase and the mobile phase differently. So as the liquid wicks up the paper, or up in this case, the thin layer chromatography and like the plate, some of the components are going to be more strongly attracted to the mobile phase, so they're going to move up the paper as the liquid moves up. Some of them are going to be more strongly attracted to the stationary phase, so it's going to stay where it is or not move very much. But in real life, they're all a little bit of both. So they're somewhat attracted to the stationary phase and somewhat attracted to the mobile phase, but they're going to move up the stationary phase at different rates. So by the end, once the red has moved all the way to like the top, so the mobile phase is now up to here, okay, our mixture will have moved, through that whole paper. Let's say, again, there were 3 things. So there's 1, 2, 3 in our mixture. Well, they will have spread out. So just by looking at this, we'll be able to tell how many things are in the mixture. So qualitative, how many, sort of, but we want to know what. So the way to test this qualitatively to know what they are, so you have to have a general idea of what they could be, and then you test those things of what they could be. Let's say that I think that one of these is iron. Okay. I think one of these things is iron. So I would take pure iron or an iron solution, and I would do the exact same test right next to it. So while this happened, and let's say that the iron at the end of this looked like this. Now I guess it should be the same color, but it went to the same location as that middle dot in our unknown thing. So that's a pretty good indication that, hey, I think maybe both of those are iron. I know that this one is iron because what I tested was pure iron. This one was my unknown. U for unknown. So then we can figure out what they are one thing at a time. So that is how qualitative chromatography works. We kind of have a visual. Now, we'll talk about how to be a little bit more scientific with this, because at the end of this, you can't be like, well, I looked at it with my eyeballs and my eyeballs said it's iron. Like, you need some sort of evidence. And so we'll talk about that when we get into the calculations component of this. If we're going to be doing it quantitatively, like we will be for gas chromatography, then it can't just be that it moves along a thing. There has to be something that at the end, when the component is separated, that there's a way to test it. So we're not going to be doing HPLC, which is liquid chromatography. But with liquid chromatography, we often test it by measuring the absorbance of the sample, like it absorbs light. With gas chromatography, we're going to light stuff on fire. So when it comes out of the end of the stationary phase, like when the mobile phase moves all the way through the stationary phase and your sample moves all the way through the stationary phase, when it comes out of the end, so like if this were, it's like when it goes out of the tops, like once it leaves the stationary phase, there has to be something right there to test it. And in gas chromatography, it's a fire and it burns it. And then based on how that happens, the instrument is able to make measurements about how much of it is. We're not going to get into details on how exactly a gas chromatography instrument works, but for any of you that have to go on or want to go on to take quantitative analysis, which is a course that is offered in the fall at St. Charles Community College. It's analytical chemistry is what it is. You get to learn more about all of this gloriousness. Okay, so that's kind of the background, the idea of what we're doing here, what chromatography is. So let's talk through calculations. So with part A, you're not going to be able to do any math until your samples that you prepared in the little GC vials and they went in the instrument, until those have been tested by the instrument. And so when you're done with that, you're going to get 4 what are called chromatograms. You made 4 solutions. So you're going to get 4 set of results, basically. And these chromatograms are going to have on them, they're going to be papers that have a graph, and then underneath the graph, there's going to be a chart. Okay, so in the graph, it's going to look something like this. There's going to be a line that kind of makes these spiky things. Okay, those are called peaks. Okay, and the peaks are a visual representation of how many components there were in the mixture, and the taller the peak, the more of that chemical there is. So in this picture, I would say that there are 3 chemicals in my mixture. Okay, chemical A, B, and C. Okay. By looking at this, I don't know which one's which, okay? But I know that there are 3 of them. And down at the bottom, what there's going to be is a chart below that has, like I guess I shouldn't call A, B, C, let's call them 1, 2, 3, because that's what the instrument does. It numbers them. So 1, 2, 3. I guess there's 3 columns. And in one of the columns, it's called the retention time. And that's because the x-axis on this graph is in time. So it timed, like started its own stopwatch. The moment that the sample entered the stationary phase, it timed how long it took for it to leave the stationary phase. So that means sample 1 left the stationary phase quickest, sample 3 left the stationary phase slowest. So sample 1 was more attracted to the mobile phase because it was able to move through the stationary phase really quickly. Sample 3 was more attracted to the stationary phase. It moved very slowly and eventually got out. Okay, so that retention time, the amount of time that it takes, is something that is, if we tested this solution again on the same instrument with the same everything, we should get the same time. So it's kind of a way to identify. And then the other one, it's the area, and it's the area under the curve. If you've had calculus, that's a thing that you've heard of before. If you haven't, that's fine. But what they did was they calculated how much stuff fits under those peaks. Okay, the bigger the area, the bigger the peak, the more of a chemical there is. Okay. So the instrument's not going to tell you what the chemicals are. It's just going to label them 1, 2, and 3, give you a time, and give you an area. So you're going to get 4 chromatograms. So that's just an example chromatogram. So let's go with, here's our 4. Graph. I'm not going to draw the tables. 2, 3, 4. And yours are labeled U, T, C, X. Okay, the U is the pure unknown. And you do have 3 components in this one, so I'm making this up. I'm going to say that's what my U looks like. And then my T chromatogram, what we did was we took our unknown and you deliberately added extra toluene. So what we're going to see, we're still going to see 3 peaks because there were 3 components in the unknown and the unknown's there. But what we're going to see is whichever peak is the toluene peak, it should be bigger in the T chromatogram than it is in the U chromatogram. So let's say this is, oops, I'm not even what I meant to do. Okay, so 3 peaks. Now, if I look at the U and I look at the T, the first peak, so peak 1, peak 2, peak 3, peak 1, peak 2, peak 3. Peak 1 looks pretty similar to me, peak 2 looks pretty similar to me, but peak 3 got bigger down here. So that's telling me peak 3 is bigger because I added extra toluene. So peak 3 must be toluene. All right, so then I have my C chromatogram. Peak 1, peak 2, peak 3. All I'm comparing is the U and the C at this point. So the C chromatogram had the unknown in it, plus it had extra cyclohexane. So we're trying to figure out which of these 3 peaks is the cyclohexane. So what we're looking for is, well, peak 1 or peak 2 or peak 3, one of them should be bigger in the C chromatogram than it was in the U chromatogram because we added extra cyclohexane. Looking at this, looks like peak 2 is bigger to me. So then I'm going to say, well, I now know peak 2 is C, which is cyclohexane. Now for X, we could kind of process of elimination, figure out what's going on here. The only peak we haven't circled yet is 1, and it got bigger, so that will be X. So the point of the T, the C, and the X chromatograms are just to help you figure out which peak is which. Okay, so what I would do is, like, we figured this out. We figured out peak 3 is toluene. So I'd go back to my U chromatogram, and above peak 3, I'd write like a T, like to remind myself. We said peak 2 was cyclohexane, so I'm going to write a C right there. And peak 1 is the P-xylene there. Okay, awesome. So T's job, the T, the C, and the X, their entire purpose is just to help figure out which one is toluene, which one is cyclohexane, and which one is xylene. We're not actually using their tables or their numbers or anything. Their entire purpose is just to figure out which peak is which in the U chromatogram. So looking on the data table for part A, the first thing you have to write down is your unknown number or letter. Okay, so hopefully you did that in lab. And then then it says retention times of internal standards from the U chromatogram. So we're looking at the U chromatogram, and there's going to be that table, right, that has 3 columns. That'll be 1, 2, 3. There's going to be time. There's going to be area. Well, and then, so what it says there is, number 1 says toluene, and then it has a blank for minutes. So we're looking at the time. So we said, I'm not telling you this is the correct order. I'm not at all telling you this is the correct order. You have to look at your own data and figure it out. But in my pretend scenario, I said peak 3 was toluene. So I would go to peak 3's time, and I'd write that down in the toluene spot. Next, it says cyclohexane. Well, we decided in my pretend scenario that cyclohexane was the second peak. So peak 2's time is what I'd write down next to cyclohexane. And then P-xylene we said was peak 1, so we'd write down its time next to that. So you're not calculating these times. You're writing them down from the table. What you're doing by writing down those numbers is you're proving to me which chemical you think is which peak. Okay, you're not mathing. You're taking the time from the table of the U chromatogram. Screw the tables on the other three. We don't need them at all. Okay, and you're just writing those down. So below that, it says peak area from the U chromatogram. We're going to do the same thing. Where it says toluene, well, we said toluene is peak 3, so you're going to write down the area from peak 3, cyclohexane. So you'll have to look at your own data, right? Now, there is actually a last line here where there's no time, but there is a total area. So there's a spot for you to write down the total area. And then you have to do the percent component calculation. This is the only calculation that we're doing for this entire part. So for the percent component. So percent, we're going to do it. All you're doing is you're going to do this for like toluene. So percent toluene. You're going to take the area of toluene divided by the total area times 100. And you're going to get yourself a percentage. Now, you're going to have a decent number of significant figures that were given to you on the areas. So your percentage needs to have the correct number of significant figures. So look at your area of your T and your area of your total. And then you're going to repeat this calculation for cyclohexane, for p-xylene. So you're going to have 3 percentages, which are giving you the amount of each solution or each chemical in the mixture. So every unknown is going to have its own percentage. All the unknowns have toluene, cyclohexane, and xylene. They don't all have the same amount of those chemicals. So again, you can't do any calculations for part A until you've been given the 4 chromatograms. You will have to turn in the chromatograms as part of your lab report because I need to see what you see when you did this. Okay. So for parts B, C, and D, which I'm going to just write down part D because that's where you're instructed to collect the data. Okay. That's going to come from your TLC plate. Okay. Your plate that's going to be the rectangle that had the line, the 1, 2, 3, 4, 5 spots. And you have your acetaminophen, your aspirin, your caffeine, your unknown something, and unknown something. So unknown B and unknown D, whatever your unknowns are. Okay. And you're going to have your line across the top, and you'll have your spots for your acetaminophen, your aspirin, and your caffeine. I'm not saying they're going to look like this. They're going to look like whatever yours look like. Okay. And then you're going to have your unknown, which might have 1 spot, or it might have 2 spots, or might have 3 spots, depending on what your unknown is. So during lab, you were supposed to measure the distance between the starting line and the middle of each spot. So you will have the distance already measured before you leave the lab using a ruler, and you will also have measured the solvent distance. So in millimeters, where you wrote everything down you knew for sure, and then you estimated one place between the tick marks. So that wasn't so much math as it was just making measurements. Now, the RF value. So you probably, with your eyeballs, were able to make some judgments here. Like, so for unknown B, that spot right there looks a lot like that spot. And that spot's acetaminophen. So it kind of looks like unknown B is acetaminophen. Okay. Unknown D kind of looks like that spot looks like that one, which is aspirin, and that spot looks like that one, which is caffeine. So it probably looks like it has aspirin and caffeine in it. And again, that's just by you looking with your eyeballs. But scientists, we can't just say, hey, I saw it, and so I know that it's real because I saw it with my eyeballs. We need some sort of evidence. And that's what RF values are. So the RF value is a calculation we're going to do for each spot. And you're going to take the spot distance, so how many millimeters you measured for that spot, divided by the solvent distance, which is the bottom to top distance. So it's the ratio of how far did the spot travel compared to how far the mobile phase traveled. And you're going to do this calculation, and you're not going to leave it as a fraction. You're going to convert it to a decimal, and you're going to get some number. Okay, so I'm going to pretend like the RF value for AC, when I did my math, I'm going to say it was 0.17. And then I'm going to say I did the RF value for unknown B, which to me looks like they're about the same. And I got 0.19. Oh, so it's like, okay, so the data value, the RF value for acetaminophen and the RF value for the unknown B are very, very similar. So that's evidence that I think unknown B is acetaminophen. And then you're going to be able to do this for all of the other ones. So this is our evidence. Now, you'll go to the correct number of sig figs. I'm not saying it's 2 sig figs. You'll have to do that yourself. You do have to show the RF calculation for each of the values. There's not a lot of math in here, so you get a point for showing your calculation. So that means if you don't show the calculation for each of them, you're going to lose a point for each of them. And that's just easy points, my friends. So show your calculations. So number 7 and number 8. It's number 7 says components in unknown blank, and in the blank you're going to write unknown B, unknown D, unknown A, unknown C, whatever your unknown is. So like for number 7, I'm going to say unknown B. In my pretend world, that's the one I tested. So in the blank, I write the unknown number or letter, and then below that, I'm going to write what chemicals I think are in the unknown. So for this one, I'm going to say acetaminophen. Okay, and then for number 8, it'll say components in unknown, and then I would write D. And then below that, I'm going to write what things I think are in it. The things you think are in it are going to be acetaminophen or aspirin or caffeine. You could be 1 or 2 or 3 things could be in your solution. So then you're going to go to the next page where you're supposed to get it from your partner group, and you're going to redo everything. Okay, everything works the same way. The distances should have been measured in the lab, and then you're calculating RF values for all of them, showing your calculation, and then deciding what chemicals are in each of the unknowns and recording that. For this first lab, there are no post-lab questions, so you don't have to worry about that. But you do have to attach or submit your chromatograms along with this. So you're going to do all these calculations that we've talked about in this video. And then you'll turn that in along with your material sheet, along with your chromatograms. And that's your lab report for this lab. So definitely let me know if you have questions. But otherwise, I think we're set up for our chromatography lab.